I'm Bonnie Bassler. I'm a professor at Princeton University in the Department of Molecular Biology. I'm a Howard Hughes Institute investigator, and I study how bacteria talk to each other. What we now understand is that the primary reason bacteria are so successful, right? They're the predominant form of life on Earth. They can make us healthy, they can kill us, they've occupied every niche on Earth. How did they manage to be so successful? And one of the ways is that they perform collective behaviors. So quorum sensing is this bacterial communication mechanism that allows bacteria to track their neighbors and to understand when they have the right number of cells that they all do something together. Whether it's good or bad, from our perspective, right, it will be successful. So that's quorum sensing. It's chemical communication among bacteria that allows group behaviors to happen. So first we get that disease-causing bacteria have to have quorum sensing to be disease-causing bacteria. So that's medically relevant. And then the sort of more, maybe more interesting part of that is if we could make bacteria so they can't talk or they can't hear, right? Then they can't count, they don't know if their neighbors are around. Maybe those would be brand new kinds of therapies that we could use to treat globally important bacteria or pathogens, including ones that are resistant to commonly used antibiotics. And so the idea is that maybe we could do behavior control by manipulating quorum sensing and making these bacteria miscount. And those could make both products for industry, but also medicines. That's a dream, but the field's getting really close now. Thank you. Oh, well, that was nice. So good evening, and thanks for coming. I'm so delighted to be here and to get to give a chance to tell you about some of the work my gang's doing on the East Coast back at Princeton. And I just want to thank my hosts, especially. It's just been an absolutely magical day for me to get to be here and to get to meet you and tell you about our science. And so I'll do a slightly better job of explaining what I do, right? Well, I should say that more accurate job. Anyway, so I'll tell you the goal of the talk. There's only one, okay? And my goal is to try to convince you that bacteria can talk to each other. And if I manage to succeed at that goal, a secondary goal is to try to show you that they're multilingual. And then the third thing that I'd love to come away with tonight is for you to think that it's actually practical to study how these bacteria talk to each other, right? Because then maybe we can think of new ways to make beneficial bacteria more beneficial and to stop harmful bacteria. So that's sort of the arc of the talk. And with that as the backdrop, I will get right into it. And so I know that this is a very diverse audience, so I want to make sure that we are all on the same page about these critters that I'll be telling you about. And so this is just a cartoon of a sort of generic, what a generic bacterium would look like for example, an E. coli that lives in your gut. And so you know that bacteria are microscopic organisms. You can't see them with your naked eye. You have to look at them under a microscope. And if you did that, you would see something that looks kind of like this, which is they are single-celled organisms, and they're covered with a membrane, which is like their skin. So that keeps the outside world out and the inside world in. And then inside the bacteria is the cytoplasm. It's the goop that has all the biomolecules roiling around that make the bacteria alive. Right? And then what's special about bacteria is that all of them have just one piece of DNA. So that's the compound that has the blueprint, it has the genes and the blueprint that makes life possible. And so they only have one piece of DNA and they have a few thousand genes, typically about a couple thousand genes. And just for reference, you have about 25,000 genes. Okay, so bacteria are the oldest living organisms on Earth. They've been on the Earth for 4 billion years. They look very primitive and simple. And if you actually watched them under a microscope, what you would see is that they consume nutrients from their environment. They grow to twice their size. They double all of these components. They cut themselves down in the middle, and one cell becomes 2, becomes 4, and so on. And so it looks like a kind of boring life that they simply eat and divide. And what's been interesting about bacteria is that they've been known for almost 500 years. We've known about bacteria. And they've always been thought to be these asocial recluses. And what I mean by that is that when they divide in half, each bacterium has no knowledge, if you will, of its sibling. They just go out and they act as individuals. Right? And what I want to try to tell you tonight is that there's no way the bacteria could be so powerful and do all of the things that I'm going to try to convince you that they do, some of which you already know, if they simply acted as individuals. They're too small to be able to accomplish these gigantic tasks that we know that they do. And so what I'll try to tell you is that the way that they accomplish that is by communicating and acting in groups. Okay, and so I want to make sure that, well, you're here, so you're obviously interested in science, but I want to make sure that you're really interested in bacteria. And so before I get into how we know these things, what I want to do is try to give you the Bonnie Bassler view of the world. Okay, and so what I've done is that I've made a picture of a man, of a person, and I used a metric, and every one of these circles represents some of the cells that make up the human body. And we know that there's about a few trillion cells, so that's how many cells it takes to give you your physical characteristics and to make you alive. And what we know about bacteria is that every moment in your life, there are 10 times more bacterial cells than human cells in you or on you. So by this metric, you're only 10% human. The other 90% of you is bacterial. But of course, it's the 21st century. There's a big helix outside, right? And so it's not just about the cells anymore. So now we know about the genome. We know about the human genome. It's been sequenced. So I remade my man in the famous A, T's, and G's and C's that make up the double helix of the DNA. Right? And of course, I already gave this away. We know how many genes roughly there are in a human being, so that gives you your physical traits and all of the other characteristics that make you human. There's about 25,000 genes in a human. It turns out there's 100 times more bacterial genes in these cells that are in you and on you. And so if you want to use this metric, you're only 1% human. The other 99% of your genes come from bacteria that are living with you as your 24/7 partners, and they are keeping you alive. And so these are not passive riders. What we know is that these bacteria that live on every other higher organism give them all these genes and proteins and functions that our own higher genomes don't have. So what is really clear now is that there would be no life on Earth if it weren't for these bacteria because of these partnerships that we live in. And so Alejandro already told you they're invisible, so we don't think about them that much, but they are with you every moment of your life, and they are keeping you alive. Okay, so that's sort of my view of the world, and we'll come back to that at the end, but I don't want to be a Pollyanna, right, is that what you do know about bacteria, and they always get bad press, because we also know, and have known for 150 years, right, that bacteria, some bacteria, when they get in us or on a plant or on an animal, they can make us sick or they can kill us, right? So historically, we have known that bacteria cause disease, and these are just pictures of bacteria taken under a microscope of some famous notorious killers. Okay, so historically we've known that bacteria can kill us, and then increasingly, going back to what I just showed you on my last slide, what we're just now learning is about these bacteria that live with us and give us health. And so, for example, we know that bacteria digest some of our food, so if you eat plant material, if you eat a salad, you don't have the enzymes to get the nutrients from the salad. Bacteria in your gut digest it, and they give you those calories. They make many of our vitamins and micronutrients that make our own biocomponents work. And then we know that very early in life, they work hand in hand with your immune system to tell it to let the good bacteria in and to keep the harmful bacteria out. Okay, so now what we get is bacteria sort of have these two personalities. They have this terrible personality where they can kill us, but we also know that they have this life-giving personality that's also essential. And so those are facts. And it's actually not what my lab is interested in. What we're really interested in is whether you want to think about the beneficial things that bacteria do or the harmful things that bacteria do. We're interested to understand how can they do any of those things. I mean, they're so small, they're microscopic. So how can some bacteria kill us and others keep us alive? And that's where we're going tonight. What I'll try to show you is that they act in teams to carry out tasks that they could never accomplish alone because individually they're too small. Okay, and so that's where we're going. And I wanted to, since this is a special talk, sort of tell you about the science path that got us to this understanding that bacteria can communicate and work in groups. And the reason I want to tell you that story is because I think it's a really good story about how science works, because this idea of bacteria communicating comes from a really crazy, anomalous place. It comes from this animal, which is called the Hawaiian bobtail squid. And so first I'm going to tell you, that's where my field started. I'm going to tell you about this animal, and then I'll get back to the bacteria. So this squid is about this big, full grown, and it's called the Hawaiian bobtail squid because it lives just off the coast, sort of in shallow, knee-deep water off the coast of Hawaii. And it lives in a beautiful one-to-one symbiosis with a bacterium which is named Vibrio fischeri. So the squid and the fish and this Vibrio fischeri, they live in a partnership that I'm going to tell you about. So first I'm going to tell you about it from the squid's perspective. So this squid is nocturnal, right? And so during the days, it goes to sleep in the sand, and then at night it comes out to hunt. Okay, so what we've done is we put this squid in an aquarium, right? And the lights are on, so it thinks it's day, and so it looks kind of spastic, right? And that's because it's not meant to be out in the day. If it was out swimming in the day, this squid would get eaten by a predator. So it looks very nervous. Okay, same squid. Now we've given it sediment. The lights are on, so it thinks it's morning, and it's going to go to sleep. Thinks it's been out, that it's morning, it's been out for a night of hunting, and now it needs to escape from predators. So it's burying itself in the sand. You just saw it camouflage itself. It's thorough. You only get eaten by a predator once, right? So what you're going to see is that she's going to bury herself a couple millimeters under the sand, right? And then in a second, she's going to retract her tentacles, and now she's invisible, right? So if I showed you this aquarium, you wouldn't know that there's a squid in it. In fact, there's about 10 of them in there. Okay, so that's a fantastic strategy if you need to escape from your predators during the day, okay? The squid is invisible. The problem for the squid is when it emerges at night and it has to hunt. So I already told you it just lives in this sort of shallow, knee-deep water. And so the problem is if the squid is swimming around at night in this shallow water, the starlight or the moonlight can penetrate the depth of the water the squid is swimming in. And so if it's swimming around, it can make a shadow. And so then predators can calculate its trajectory, see that shadow, calculate its trajectory, and eat it. So the squid has to have some kind of way to be invisible at night. And this is where this symbiotic partnership with this bioluminescent bacterium that I told you about named Vibrio fischeri comes into play. So now you're looking at the squid. It's been turned on its back. And I hope what you can see are these blue glowing orbs. So what the squid has evolved is a specialized light organ. It's under the mantle or the body of the squid. And in that light organ is this bacterium, Vibrio fischeri. It's like at 10 to the 12th cells per milliliter. It's like toothpaste of this bacterium. It's living in there, and this bacterium has the special property that it's bioluminescent. It makes blue light. And so what happens is when the squid emerges at night to go on its night of hunting, it's got detectors on its back, so it can sense how much moonlight or starlight it's hitting its back, and then it opens and closes a shutter. It's just its ink sac under this light organ, which modulates how much light is coming out made by the bacteria. So it senses how much light hits its back, and then it allows the same amount of light to come out of the bottom as is hitting the top, and so it counter-illuminates itself. Right? And so by using the light from the bacteria, it has this anti-predation device that again makes it invisible because it doesn't make a shadow. Okay, so the squid is willing to feed these bacteria and house them in this light organ because it gets this anti-predation device. The bacteria are happily living in there because this light organ is all full of nutrients, and it's a much richer, fatter, happy life than trying to fend for yourself free living in the ocean. Okay, so now you get it from both the squid and the bacterium's perspective, but what's interesting is that the bacteria only make light at night, right? So how can the bacteria know whether it's day or night? And now what I'll do is switch to the bacterial perspective and tell you it's because they're talking to each other. All right, so now we're gonna go to the bacterial perspective. So this is supposed to be a cartoon of a bacterial cell. So the world is big and bacteria are small. So when a Vibrio fischeri cell finds itself dilute or alone, it wants to have the program of gene expression that's good for acting as an individual. And in this case, that means don't make light, because one bacterium can't make enough light to make perceivable light. But what the bacterium does is it makes and releases small molecules that I've depicted as these triangles that we call autoinducers, and they're like hormones. But what happens is, to remember, if the bacteria are alone, it makes and releases these autoinducers. They diffuse away from the cells, and the bacteria can't detect them. That says, you're alone, don't make light. But then as the bacteria grow and divide, since all of the cells are making a share of this hormone, this autoinducer molecule, the amount of that chemical outside of the cells increases in proportion to cell number. And when that molecule hits a particular level, the bacteria detect it, and they infer from that detection event that they must have neighbors around. So they're using the concentration, the level of the chemical, as a proxy for cell number. And when they detect the molecule, all the bacteria together turn on light. And so since they're all together, they make perceivable light because there's a lot of cells together, and that's useful for the squid. And so the reason that they only make light at night is because what I didn't tell you is that the squid has a pump. So this, you can imagine, this is that light organ filled with bacteria. They're making light. The squid is using it to escape from predators. But then when the squid goes to sleep in the sand, the way I told you, it has a pump that's attached to its circadian rhythm, and it pumps out like 95% of the bacteria from that light organ. So now the light organ is like this. So over the day, while the squid is sleeping, the bacteria have been diluted. They start growing again. This molecule accumulates, and then at night, the bacteria are in this configuration, and so they all turn on light exactly when the squid needs it. So the squid is modulating this by pumping the bacteria out each morning. Right? And we know that we're right about this because we can do all of this in a test tube, and we can spin in a centrifuge. We can spin all the cells out of the solution, and if we take the liquids, right, that were left over, they have the molecule in them, and if we squirt them on these cells, they will turn on light. So in fact, the bacteria have no idea how many cells are around them. They depend on this chemical to build up, and they use that to infer that there must be neighbors, right? And so it's a way of communicating with chemicals. Okay, so discovery was made, and then of course we wanted to bring the tools of molecular biology to the table and ask, how does this really work? What are the components involved? And so what we did by using genetics and biochemistry was to find the components that make up this little chemical communication circuit. And so now this is supposed to be my Vibrio fischeri cell, and what we found was that there was a protein, an enzyme, that makes that hormone, the autoinducer molecule, and then that just diffuses out of the cells. So the more bacterial cells there are, the more of the molecule there is. And then there was a partner protein. It was called a receptor, and it sits on the bacterial membrane. And there's a part of it that's on the outside and a part of it that's on the inside. And this is exactly like the hormone receptors that sit on the surfaces of your cells. And so what happens is that this receptor has sort of a slot in it that allows this autoinducer, when it gets to a high amount, the autoinducer can bump into this receptor. And when the autoinducer slots into the receptor, that alerts the receptor to send information into the cell to turn on the genes that make the enzymes that make light. And so it's really simple how this works. The more bacteria there are, the more of this autoinducer hormone that there is. At high numbers of cells, which is high numbers of the hormone, information comes in and all the bacteria turn on light together. And so they can do this in synchrony by measuring the concentration of the chemical. And so the reason that that is interesting, beyond just being interesting, is because we found this originally in these bioluminescent bacteria. But once we got onto this idea, we were thinking, well, can this be the only example of it? And sure enough, it turns out that this is very general. At last count, there were 14,000 examples of bacteria that all have, all kinds of different bacteria that have these chemical communication circuits. And so what I mean by that is they each have an enzyme that makes the hormone. There's always a partner receptor. And then together, these complexes turn on all kinds of behaviors that bacteria want to express when they're in a group, but not when they're alone. And so now we have a fancy name for this process, for this field. We call it quorum sensing. And so the bacteria are sort of voting with these chemical votes. The vote gets counted. They decide if they have enough neighbors present to do something that if there were fewer of them, that task would not be successful, right? And so in every bacterium that's been studied, there's hundreds and hundreds of genes. This is much more than being about bioluminescence. There's hundreds and hundreds of genes that bacteria turn on as groups that allow them to do these collective behaviors. So that's what we call the process now. And then these are not triangles and squares, right? We actually know a lot about these molecules as well. And so now I'm going to show you some of the molecules. And so in my cartoons, they're depicted by these triangles, but this is the actual chemical, that's the word that the bacteria talks with. And so, of course, the first one that was figured out was from Vibrio fischeri, this bioluminescent bacteria. And so the chemical that's used for communication is this ring with this chain. These are just carbons. And then we and other scientists started to look into all these other quorum-sensing bacteria and ask what are the chemical words that they're using. And so on this slide, I just have a smattering of different quorum-sensing bacteria and then the molecule that each one of these bacteria uses to communicate. And what you can see right away is that all the molecules are related to each other. So all of the molecules have this ring on the left side, but then these little carbon tails are a little bit different in each species of bacteria. And so what those tiny differences in the molecules do is to confer exquisite species specificities to these languages. And so what I mean by that is if we take the Pseudomonas molecule and we put it on Vibrio fischeri, nothing happens. And likewise, Pseudomonas is impervious to the Vibrio fischeri molecule. So see how they kind of look like keys? They really fit like locks and keys into their partner receptor. And so what I mean by that is that the Pseudomonas molecule fits into the Pseudomonas receptor. The Vibrio fischeri molecule, like I already showed you, fits into the Vibrio fischeri's receptor, and there is no slop in these systems. Right? And so what we understand from that finding is that these molecules enable within species communication. This is how bacteria talk to their siblings. These are private, secret conversations that they and their kin have that allow them to understand when they have lots of their kin around and they should take on some kind of task as a group. Okay, so that's the molecules, and we also know a lot about these behaviors, like what are the kinds of behaviors that bacteria control as collectives. And so I told you in detail how Vibrio fischeri uses quorum sensing to control turning on and off bioluminescence. Right, so again, these are just some of these different bacteria that we now know have quorum sensing communication, and in lots of other cases that we have studied and other scientists, what bacteria do as a group is that they make a biofilm, which is how bacteria sit on surfaces. So they make a film. The one you know about is on your teeth every morning. So they make a film. That's the first step in an infection. And it takes a lot of bacteria to make that community. And then together, they all secrete their virulence factors, which are like the toxins, the poisons, the things that actually make higher organisms sick. And so what we now understand is that in many clinically relevant bacteria, if they don't have quorum sensing, they're not pathogens, right? So they have to do these tasks as groups. And so I'll just give you one example. Pseudomonas aeruginosa is a notorious pathogen. It just lives in the dirt. You and I encounter it every day, but it's an opportunistic pathogen. And so what happens, so it doesn't hurt us, but if a person has cystic fibrosis, which is a disease that people have that is a genetic defect in their lungs, they can't clear their lungs, what happens is typically when a person is in his or her teens, for reasons that we do not understand, they become permanently colonized with Pseudomonas aeruginosa, and people with CF die from a Pseudomonas aeruginosa infection. Right? Pseudomonas is also notorious in hospitals. It likes to make biofilms on surfaces. It infects wounds, burns, things like that. And so what we know is that in all of those contexts, Pseudomonas has to have quorum sensing to make those infections. And so if you think about it from the bacterium's point of view instead of the human point of view, this is a fantastic strategy. So your immune system evolved to hunt down invading bacteria and get rid of them. So the last thing a pathogen wants to do, if there's just a couple bacteria in you, is to start secreting its toxins. First of all, one bacterium can't dribble out enough to make an infection. And second of all, those are red flags for the immune system. So the better idea from the pathogen's point of view is to wait, to count itself with these small molecules and recognize when they have the right cell number that if they all launch these attacks together, they will be able to colonize a much more enormous host. And so what we get now from our studies is that if you make mutants, if you make bacteria that can't talk or they can't hear, they're not pathogenic, right? So, and we will come back to that as we go along. All right, so that's quorum sensing. And I think the way to sort of generally think about it is that whenever a bacterium is going to give something away, a secreted product, it never gets its own back. But if I make it and you make it together at the same time, I get the benefit of your work and you get the benefit of my work. So it's sort of about public goods. The bacteria put something out in the world and they have to get their neighbor's product back. That is quorum sensing. All right, so once we got that far, we started to think these bacteria are chit-chatting, but then we started to think more about how bacteria really live in nature. Like, it's great if you're Vibrio fischeri and you have this pristine environment and you're the only bacterium in this light organ of this squid, but that's actually kind of an exception. Most bacteria live in mixed species communities with hundreds or thousands of other species of bacteria. And so what we started to think is that if quorum sensing is so specific, this within-kin communication, how do bacteria manage this when they're living in mixtures? Okay, so here's a picture of a bacterial community. This is a biofilm. This is a picture of human elbow skin under a microscope. So this is what your skin looks like. It's covered in a biofilm. And the point that I want to make about this picture is that every one of these different shapes and sizes of bacteria is a different species, right? So you have something that's estimated like 600 species of bacteria on your skin. And so the question that we started to wonder at this point in the studies is, how do bacteria manage to count if there's all these other species around? And so of course we didn't know the answer to that, but we wanted to think of a way that we could explore that question. And so what we turned to was a cousin of Vibrio fischeri. So we knew about a bioluminescent bacterium from the ocean that's named Vibrio harvii, and this bacterium makes light, like Vibrio fischeri. It only makes it at high cell density, so we knew it had to have quorum sensing. But what's really neat about this bacterium is that it lives free living in the ocean. So it's a very common bacterium in the ocean, and we knew that it lives in consortia with all kinds of other species of marine bacteria. And so we thought maybe its quorum sensing system would be more interesting or more complicated to study because it had to navigate a world where there were all kinds of other bacteria around it. So we wanted to start studying this critter. And the reason to show you this slide is actually to try to show you something about some of the experiments that we do in the lab. So what you're looking at is just a flask with Vibrio harvii. Here's a few Petri plates, some test tubes, obvious graduate student propaganda that, from my lab. But the point that I want to make is to take this picture. All we did was turn the lights off in the room. We're not doing anything to the cells. We took the picture with the light that the bacteria are making, right? And so I've already alluded to this, right? We can see this with our eyes, right? These bacteria, by making light, have made their invisible world visible to us, the scientists. And so what we could do here, which is similar to the Vibrio fischeri story, is we could simply make mutants, just make mutants in the bacteria, plate them out on these Petri plates, go in the dark room, and look for bacteria that were dark when they should be bright, or bright when they should be dark. And just by doing something so simple, just turning the lights off in the room and looking for bacteria that were goofy in the way they made bioluminescence, is how we found the Vibrio fischeri components and how we're going to find the ones for Vibrio harvii. So it was really simple, and we exploited this natural readout of the bacteria talking. When they talk, they make light, right? So we looked for defective bacteria in that process. Okay, so here's what we found. So this is Vibrio harvii now. What we found was, sure enough, there was an enzyme that made an autoinducer. I actually showed it to you on a couple slides ago, and there was its partner receptor. So that was very similar to the Vibrio fischeri system. But to our surprise, when we eliminated this quorum sensing system, this bacterium could still sense its neighbors. And so what we found was that there was a second system. So there was a second enzyme that made another autoinducer molecule. It also had a partner receptor on the membrane. And then together, the information from both of these systems comes into the cells, the Vibrio harvii cells, and tells the cells to make light and hundreds of other things that they do at high cell density. But in our experiment, we're just measuring light. And so what we could see was that there had to be two quorum sensing systems. And so once we found those components, we started to wonder, so why would this bacterium be built this way? What's the point of having two quorum sensing molecules? And our idea was, if these two molecules don't encode different pieces of information, then having two molecules isn't better than having one. And so we already thought we knew what this molecule was for. This is for within species communication, right? This is the intraspecies communication system. So we wondered what this one was for. And so what we did was we collected up every bacterium that we could find, and we spun the cells out. And remember, the molecules are on the outside. So we spun the cells out in a centrifuge, and we collected up the fluids, and we looked for molecules. And lo and behold, what we found was that every bacterium we tested, every bacterium made this molecule. And so we purified this molecule, we figured out the structure of this molecule, and this is it. It's 5 carbons, these are carbons, and it's decorated by all of these oxygens. That's actually not the important, this molecule is very dear to me, but that's actually not the important point. The point is, no matter what bacterium we purified the molecule from, all of them made the identical molecule. They made only this molecule. So unlike the last language that I told you about, where each species has sort of a different dialect, in this case, all of the bacteria were using the same molecule to communicate. And so our interpretation of that result is that this molecule isn't species-specific. This is sort of the trade language or the Esperanto of the bacteria. We think this is the molecule that bacteria use to talk across species. And so now what we think is that bacteria have to be at least bilingual. There's probably more molecules to find. And that in fact, in general, they're set up like this, where they have one molecule and one system that allows them to count their kin. And then they have a second system, this generic system, that allows them to communicate across species boundaries. And so what we've shown that they do is that they actually measure the ratios of these molecules. So here's the computation we think bacteria do. So bacteria are just out there, and what happens is that they are scanning the environment for quorum sensing molecules, right? So they're trying to understand, if you will, am I alone or am I in a community? So they're scanning the environment for quorum sensing molecules to start to tell them what their behaviors ought to be. But then the more sophisticated computation that we showed they do is they measure the ratio of these two molecules. And that, we think, says, is it mostly me and my kin, or is it mostly not my kin, is it mostly the enemy? And then they change their behaviors based on whether they are around mostly themselves or mostly a different species. So we think that these molecules allow bacteria to distinguish self from other, and to do something about that. So what we've shown is that they tailor these programs at the bottom based on whether it's mostly kin or it's mostly foe. Right? So in fact, we think the idea of communication, collective behaviors, and being able to tell self from other, we think that evolved billions of years ago in bacteria. Okay, so that's how we think quorum sensing works. And of course, I promised you there should be a practical part of this, right? And then I've already told you that these systems control virulence. In pathogenic bacteria, both of these molecules control virulence. And so once we learned enough about these quorum sensing systems, we thought maybe we could think up strategies to interfere with these communication circuits to try to stop pathogenic bacteria from being harmful. And so I know everyone in this audience knows that we have this global urgent problem of antibiotic resistance, where we don't have new antimicrobials that are working against bacteria. And so people in my world, the world of people that study bacteria, we're trying to think, are there new Achilles heels that we could go after to try to make new kinds of therapies? And so maybe quorum sensing could be one of those. And so we wanted to try to put that to the test, and I'll try to tell you about some of our efforts along that line. So the idea is that we're going to interfere with quorum sensing so bacteria can't count appropriately and they don't launch these collective behaviors like virulence. Okay, and there's sort of two ways to do it. One is if you can interfere with the species-specific language. So lots of times, right, physicians, what they want is what's called a narrow-spectrum antimicrobial. So that is, you have a Pseudomonas infection. We want to have a compound that very selectively targets Pseudomonas. And so one can think that perhaps if we could interfere with these really selective systems, you could make narrow-spectrum therapeutics. The alternative is that many times a person or an animal is sick and the physician doesn't even know what you have. And so in that case, you get treated currently with broad-spectrum antimicrobials. And so maybe we could take advantage of the second system, right? If we wanted to make a anti-quorum sensing strategy that would work against many species of bacteria, then you could go against this interspecies generic system. right? And so the logic to do this is the same in both cases. You try to shut down communication through one of these circuits. And so I'm just going to show you one new experiment that is about trying to do this intra-species interference. Okay, and it's going to be with this pathogen that I've already talked about, Pseudomonas aeruginosa. So this is just a Petri plate of this pathogen. This, I already told you, is a notorious pathogen. It causes the highest number of hospital-acquired infections in the United States. It is a notorious biofilm former. So it's really sticky. It likes to stick to biotic and abiotic surfaces. And so you guys probably know now it's like, in the 17th century, you go to the hospital now because you want to get your gallbladder out and you get infected by these terrible bacteria that are multidrug resistant. So Pseudomonas is one of these that's really on the rise and that is a huge problem, right? And so we wanted to, if we were going to test this idea, we wanted to try to go after a pathogen of real relevance. So we picked this bacterium, and then also we picked it because we knew a lot about its quorum sensing system. So I showed you its autoinducer before, and what we already knew is that quorum sensing is essential for this bacterium to make these biofilms and then to produce its toxins. It's got like 50 or 60 toxins that it secretes as a group, and so we thought this is a good one to try to go after to see if we could interfere with quorum sensing. Okay, so what's the strategy? Okay, so I've already shown you the Pseudomonas autoinducer. So this is the actual molecule that Pseudomonas uses to communicate, right? And so we knew that molecule. And so what we did using chemistry in the laboratories, we made a synthetic molecule that looks like the autoinducer, but that is an inhibitor. So if you look, here's the real molecule that Pseudomonas makes. This is the molecule that we made. And so what you can see is that this part of the molecule, the left-hand part of the molecule, they're almost identical. There's a sulfur here instead of an oxygen, right? But this part, if you think about these like puzzle pieces with the receptor, the molecule looks kind of like the autoinducer, but then it's got a big bump on the right-hand end, right? And so the fantasy was that our molecule would slot into the Pseudomonas receptor and jam it so that this molecule can't be detected. And the reason we put a sulfur here is because humans, we have an enzyme that, if there's an oxygen here, we have an enzyme that opens the ring. So this would be impervious to the enzyme in our body, right? So it looks basically the same. And the idea is it slots into the receptor and it stops real quorum sensing from happening. Okay, so that was the strategy, and it was based on knowing the real Pseudomonas molecule. Okay, so we made that inhibitor, and so now I have to show you that this actually works, and so I will. All right, so remember, I've told you the two steps in virulence, right? The first is that Pseudomonas gets on a surface and it makes a biofilm. So what you're looking at here, this is a Petri plate, and what we did was we just put a drop of Pseudomonas right there, and then we incubated that plate. And then overnight, Pseudomonas crawls out over the plate to make this beautiful pattern. This is a biofilm, right? So it started as a little drop, and it grows and spreads out. Okay, so that's the first step that mimics the first step in an infection. So what we could do is we could make the same Petri plate, except we put that inhibitor molecule in. And sure enough, it stops Pseudomonas in its tracks. It just stays there at the center of the plate. It can't apparently make the biofilm. So at that point, we thought we were cooking with gas, and we were pretty happy about that experiment. And so then we wanted to move on to the second step in virulence, and that's the group-wide production of these virulence factors. So we have an assay for that in the lab. So Pseudomonas, if you noticed on that first Petri plate and then on my cartoon, I drew it as green. Because if you just grow a test tube of Pseudomonas, this is a big test tube, this is just Pseudomonas grown in some liquid, the whole culture turns green, and that's because Pseudomonas makes a toxin. It's a compound that kills human cells, and that toxin is called piocyanin, for cyan, right? And so in fact, when quorum sensing happens and Pseudomonas makes this toxin, the toxin just naturally has a color, and it's green. So here you're looking at a dense culture of Pseudomonas, right? It's grown to a high cell number, quorum sensing has happened, it's secreted this virulence factor, and the entire culture just turns green from this piocyanin poison that Pseudomonas is sort of spurting out. We can make a mutant in Pseudomonas that doesn't make the quorum sensing autoinducer, right? So if it doesn't have the autoinducer, it can't count, right? And so what you can see is that in a test tube, now the culture of Pseudomonas is colorless, right? Because it doesn't know it's at high cell density, so it doesn't make the toxin, right? So everybody gets that. So that's that is what it looks like when no quorum sensing can happen. So now what we did was we took the wild type Pseudomonas, right, and we squirted in our inhibitor and we asked what happened, right? The antagonist that shuts down quorum sensing. And what you can see is that the culture is sort of pale, pale green. What we can do if we quantify this, it shuts down the production of piocyanin by 90%. So now we think that that looked pretty good, right? That in fact, by just interfering with quorum sensing both on the surface and in the liquid, we could hopefully shut down quorum sensing. And so now, of course, we wanted to, once we got that, we were pretty confident about that result, and so we wanted to do it in an animal. So here's the last experiment that I'll show you tonight. So what we have is an animal infection model, right? So this is the percent of the animals that are alive over time. And so, of course, if we don't infect the animal with the Pseudomonas, right, all of the animals, this is sort of a day later, all the animals are alive. Right? If we give an infection with wild-type Pseudomonas, what you can see is that in a day, nearly all of the animals die. Okay, so that we knew had to happen because Pseudomonas is a pathogen of the animal. So now the question is, if we give the animal the infection, but we simultaneously give our anti-quorum sensing inhibitor, what happens? And the answer is that all the animals live. Right? So this started to look pretty good to us. I want to be really careful here. This is not a medicine. Right, you'd have to take a pill like this big and it would kill you, right? So, right, what we did, this is a proof of principle experiment, right? We wanted to see if there could be merit to this idea that, was untested, right? Before we did all the chemistry that one does to make the molecule very potent. It's not that potent. We have to give a lot of it. It's not that potent. It doesn't just go where we want it to go. It can't actually be toxic to you either. And so what happens at this step with this, what's called a lead molecule, is now we do the chemistry to make the molecule potent, to make it safe, to make it delivered where we want it to make it have a reasonable half-life. None of that has been done. Right? So I don't really know if this can work in a human yet, but I think that this kind of experiment starts to be the kinds of steps that we're taking to make us think that perhaps we should keep going in this direction. So that is the story that I wanted to tell you tonight, but now I want to just recap a little bit about what I've told you. I hope what you think by now is that bacteria talk to each other. So of course they don't have voices, and so their language is chemical, right? So they use chemicals to talk. I think what that means is that bacteria are multicellular, and so of course, right, we know about multicellular organisms on Earth, and we also know that all of us came from bacteria, right? So they were the first organisms that gave rise to everything else on Earth. And so I think that by studying this very primitive form of multicellularity in bacteria, what we hope, beyond learning about quorum sensing, we hope that we're actually learning about how all multicellularity evolved on this Earth, and that maybe the principles about how cells do things together, how they do it robustly, how they avoid cheating and free riding, right? All those things, we think that maybe by studying these bacteria, we're going to be able to help our colleagues study higher organisms because sort of the skeletons of the systems exist in these very ancient single-celled organisms. What I already told you during this talk is that with these two molecules, we believe that what those mean is that bacteria can distinguish self from other. And again, that seems like a trait that we know about in higher organisms, right? That happens all over in animals and humans. We know that our brains allow us to tell self from other, and we think, again, that that evolved in the bacteria, and that by studying bacteria, we can maybe have some insight into how that happened in other organisms. And then the practical part of this is that by learning that these bacteria have these conversations and have collective behaviors, we can develop strategies to impede quorum sensing in harmful bacteria. But then to go back to the very beginning of my talk, where I told you that bacteria mostly get a bad rap, right? The last 10 or 15 years has all been about this magical microbiome, these bacteria that live on higher organisms and keep us alive, right? We're only now learning that bacteria do this magic, right? And so in fact, it may be better for us, instead of fighting against these harmful bacteria, maybe the real way forward in medicine is to improve the quorum sensing that we know happens with these beneficial bacteria, because remember, they are fighting hand in hand with your immune system to keep invaders out. And they're mostly keeping us healthy, these bacteria that live naturally with us. And so what we're also trying to do is to make pro-quorum sensing molecules that would make the bacteria that normally live with us better at making us healthier, right? So we have a pony in that race as well. And then the last thing to do is to finish with the confession, which this whole business of like being able to interfere with quorum sensing, we didn't think any of that up. These bacteria have a 4 billion year head start on us. And so once we sort of got onto this idea that, oh, maybe we could interfere and we could manipulate quorum sensing, we thought, well, haven't these bacteria already done this already? And so we and lots of other scientists have now been sort of going out into the environment and looking at how these bacteria really live in these natural communities, not laboratory communities, but natural ones. And sure enough, they're out there, they eat each other's autoinducers, they eavesdrop, they free ride, they cheat, they try to make these antagonists. And so sure enough, they're doing all of these tricks. There's all this shenanigans going on out there. And if you think about it, the behaviors, these pro and anti-quorum sensing behaviors that are happening out there in nature have been tried and tested over billions of years, right? And so the ones that exist have to be really, really good. And so what we're thinking now is that instead of starting from scratch, like I showed you on that first experiment, we should go out there and let the bacteria tell us which are the best strategies and bring those into the lab and then tinker with the strategies that they've already done the work of creating and testing over billions of years, because those might really be the best for both medicine and applications. Right, so that is a big part of the field now going forward. Okay, and so that is my story, but I do want to tell you about the heroes because they are not me. So this is a picture from this summer of my lab and our families on my back porch, if you're interested in that. And I just want to especially tell, I see a lot of budding scientists, I think, out in this audience. I just want to tell you that it's the greatest life in the world to be a scientist, right? Your life, your job is to make discoveries, right? And think about things that, crazy things that nobody's ever thought about before, and then try to do something useful from that. And so if you like doing puzzles, if you like solving mysteries, if you like thinking crazy ideas, you should really give this a try, right? Because it's really a fun life, right? And then what else is neat is you always get to be around smart, creative people who are willing to go on this amazing adventure. And I just want to show you that these people look like you. They don't actually look like me, right? The science that gets done, at least in this country, is done by young people, right, who just want to give it a whirl and want to think that they can make a contribution by making discoveries. And so it's really fun and a privilege to get to work with them. And I'm happy at the reception if anybody wants to talk to me about that, especially students maybe, that it's just a wonderful way to spend your days. Anyway, so I want to thank you again for having me, and I think I get to answer questions if you have any. So thanks for listening.